Okay. Yango, ngarang si ekoi photo system se khangba loireba. Photo system se khangba loiramnina ngasi dagi hoduse will start the mechanism of photosynthesis hodromne. Yare ko. Now, ekoi na sarna haigi bada photosynthesis haibase. Ngasi taktoi se mamanggi class ta ekoi na nainagiba kanda photosynthesis haibasi light reaction plus dark reaction ne haibe yaine. Chumiko, photosynthesis involves two reactions. The first is light reaction. The second one is dark reaction. Yareko. Ahanbagi step light reaction set. The first step, light reaction deals with preparing the light. Sorry, preparing the energy. Yareko. Light reaction gi matamda tawdoi se kaineba energy prepared tawdoneba. Energy in the form of ATP and NADPH2 haigibdo. Anisuba step ta adugi energy to shinaraga dark reaction gi matamdana kai prepare tawdwa food in the form of glucose prepared tawdone. Yareko. Ngachi sir na taktoi Mokoi glucose sungdringaida kai hana sungsega energy hana amta sungsel. Aniko by using the first reaction called as light reaction sishinaraga. Aduri angasi aina take tawrisi light reaction. Howe aradra light reaction. Yareko mechanism hodrom ni muna takpiro tarasi maram horen noina marakta khangdaba khotaba leibado hangbiro. So let's start the light reaction. Light reaction. Kitang muna tasiko kayam nungai horen horen yum halaga pasiko kayam nungai Light reaction, which is the first step of photosynthesis involving preparation of energy using sunlight. Hence the name light reaction is given. Yareko. Now, light reaction ki matamda sungdoi se kai neba. Energy. Normally kai cell amana use the way of energy se ATP ni na. ATP neba sungdoi. Energy neba sungdoi se shi matamda. Ado akoi hana khange energy di kamai sungye ATP dia. ADP amaga kai ga punchan araga inorganic phosphate ka punjanarakandaने kai sungdoise ATP haina sungdoise yare ko adi ATP sungedi kai add toksanbam changi phosphate ama kanada addition of phosphate is called phosphorylation haina ko humro sarnai se how are we going to prepare ATP by adding one phosphate molecule to ADP addition of any reaction involving addition of phosphate is called phosphorylation reaction ko Hobatabu, light reaction ki matamda ATP sungdoise. We are going to add phosphate to the ADP and prepare ATP using light. Anasi se photophosphorylation jukoy. Light reaction se. Akoi kai hapsan done ba phosphate kanada. Adu kai sungdo ba. Only thing is that this reaction has to happen in the presence of light. Aduna photophosphorylation jukoy ba light reaction ke to means. Yare ko phosphorylation. Ai sibok tang han thagi ko. May I know what is phosphorylation? Addition of phosphate to the ADP to form ATP. Since this reaction is to happen in the presence of light, I'm going to put one more word among the get this that is photophosphorylation ko. Yare ko. So light reaction haibaga photophosphorylation haibaga chapmane. Yare ko adi photophosphorylation hairaga na kamain khumtare. Preparation of ATP using light generaga ATP sungbase photophosphorylation ko. And also we call it light reaction. Yeah, we discussed that this particular reaction occurs in light reaction it occurs in brena thylakoid natra it occurs in thylakoid membrane I did thylakoid ano ina thage ko thylakoid membrane the satay kaigino thylakoid membrane the sathari because thylakoid membrane the light trap so ngamba pigments yawagi yare ko thylakoid membrane Gyan taribro takbo hodri topic maming se gyan taribra tari ko aduna ngasi tamdoi se sarna photophosphorylation ne yani ko technically hairagana photophosphorylation common term the hairagana light reaction haina ko yare ko adudi light reaction oigera photosynthesis or photophosphorylation oigera it's all about preparing energy in the form of ATP. Kamaina prepare todova photophosphorylation se kamain todono haitari we have two mechanism. Mechanism ani lay. Photophosphorylation is the reaction involving preparing ATP, and how we are going to prepare ATP haitari. We have two mechanisms to do so. Okay, one mechanism is called non-cyclic photophosphorylation and another is called cyclic photophosphorylation. Haina ko yare ko mechanism amana kai ko ba non-cyclic photophosphorylation haibama photophosphorylation haibama. The other mechanism is called non-cyclic, sorry, cyclic photophosphorylation. The other one is cyclic photophosphorylation. Ay samjalage swain photophosphorylation. Yare ko, sina kanagi type ani no? Photophosphorylation gi type ani. Type ana hairasu ay mechanism hairasu ay ko. Light reaction gi matamda ATP kamaina shungi hena hatardi. We have two mechanism. One mechanism called non-cyclic and another called cyclic. Yaraba. So I will start from non-cyclic photophosphorylation digi tak hudane. Sibokta understand tobra? So we ko marakte any doubt later hango ko. Step ama kan gitardi lu winning ni. Non-cyclic photophosphorylation hairiba light reaction gi reaction mukal se. Masi chatha ge haitardi it involves both photosystem 1 as well as photosystem 2. Anime involved to. Yare ko. Masi gi reaction se thok na hot the photosystem kya involved to ba? It involves both photosystem 1 as well as photosystem 2. Photosystem anima involved to kana se thokna bu khotna bada non-cyclic photophosphorylation se thokna khotna yare ko noi ngarangi matem tang recall to minata di photosystem 1 kai da lei grenada sulei stromala meli de sulei photosystem 2 na kai da lei grenada adudi grenada photosystem 1 su 2 so nimak lei ne adi photosystem 1 su 2 su animak leiba mafam duda animak darkar oiba reaction se thoidra so non-cyclic photophosphorylation occurs on grena. Why? Because grena has got both PS1 as well as PS2 animak lay ne mongonda kai thoni ba non-cyclic photophosphorylation da kai da thok ba it occurs in grena da thwe ai di grenal thylakoid da thoni hangi ko khangda di sir kitang khak elaborate to yari khangda ba yawre nge khutang lo kitang hangaluro kitang halluro matim tang matanggi sahi gi September bok takshi yengosida dali ko no je ta sum haiti singarang sir chloroplast ma yeki natra chloroplast si manungda yauriba Bricks like structure mayam se kai kowen tamgiba thylakoid right when many such thylakoids are stacked upon one another it will form grana so I'm formed there so this is one granum I will draw another one what is this thylakoid 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 this is another granum yani kwa hangnaba grana anisi they are interconnected by a breeze we call the breezes stroma lameli ena noi director is stroma lameli su thylakoid ne thylakoid ki extension natra dum yengo thylakoid ne na asna intergranal thylakoid chu ko grana ani samna ba thylakoid yare ko si tu thylakoid la sida le sina granal thylakoid ko sina stroma lameli or intergranal thylakoid ko haining ba haiba yai marang tam gi basa thylakoid ki membrane da kai yaw doino thylakoid ki membrane da pigments nap to ne amamam tang no in group we call it the thylakoid ki membrane the pigments nap ni hairagum se haito male photosystem nap kani no in hairse photosystem the pigments group oina yarni ko aduda photosystem makal kya tam bige koibo ani photosystem 1 se kai waida leige di ma di kai da leyeba grena da sulai stroma lameli da sulai haine basar na haise yan ta bro sila uli se kai ne this is photosystem 1 this is also photosystem 1. So photosystem 1 is located both on grena as well as stroma lamellae. Ani magtalayba. Whereas, aniswa photosystem do kay photosystem 2 is located only on kaida grena khagtadaleba. Sina kay photosystem 2. Adoy tari grena da photosystem 1 so 2 so ni maglayraba. 1 so 2 so the car over reaction do is the kay ko hage. The non-cyclic photophosphorylation occurs only in grena the thui hai name is na isi. Bu sing sidi so me ngo kaida thueba grena da sime are ko somsing ale ko na taxi to bu comparison to na hypotang irgige cyclic photophosphorylation chathage haitardi it involves only photosystem 1 photosystem 1 khaktamak involved kai khaktamak it involves highly very animak se reaction ngakne kai prepare to never reaction ATP prepare to never reaction eva sigi reaction se thokna one of the photosystem ani chathai changi hairiba reaction se grenada to wi haibo ani ketoroge Yareko, CG reaction je tawna one of the it involves only how many photosystem? It involves only photosystem 1, PS1 khaktamak involved tayo. PS1 di kay dalay stroma lamelli de sulay, grena de sulay. So it occurs in both stroma lamelli as well as grena. Ani magtathokani ba madi. Yareko, can I put that way? It occurs in kay de tueva. It occurs in grena haigera, grenal thylakoid haigera, and kaday de sutokani ba and stroma lamelli de sutuev. Yarabo? Yareko? Asina magi thokpam aduga reaction se tawda changba photosystem dugi wari khange. Yareko? Now let's go deeper into what actually is non-cyclic photophosphorylation kaino yengminar sebasar. Yengasiko? Can I change the slide? Look here. Somde yongsiong kitang muna tamina siko. Kaigino da khara complex level reaction ni na. Sergi blackboard se. यानी को ब्लैक स्क्रीन से यारे को थैला कोई टी मेम्ब्रेने हैना लोअर से ग्रेना आगे स्पेशली करम करम फोटो सिस्टम नप्पल रमने फोटो सिस्टम 1 सु लेरमनी फोटो सिस्टम 2 सु लेरमनी बको ए फोटो सिस्टम 2 दगी हाना होरके यानी को सी फोटो सिस्टम 2 ने लोअर से once again may I know what is a photo system? It is a group of pigments at the center of the group there should always be okay so if I'm saying this is photo system at the center we should have chlorophyll A. Panagineva Sarni say I'm showing you photosystem 2. Yarabo, what is this called? Reaction center. Surrounding the reaction center are the accessory pigments. Very good. Are the accessory pigments. Yaudone. Kongra. Yareko. Si akoy basina kay ngarang tam po ne basili. Akoy basina kay accessory pigments. Mayay day sina kay chlorophyll A. Marak tayo na point namtang hapo basa. Kanano accessory pigments Chlorophyll B, carotenoid, xanthophyll. Can chlorophyll A be accessory pigments? Answer is yes. Yani ko mayayda yawrido reaction center kaujagibwa neba. Reaction center oine act tawdaba singduna dum accessory oine laigini. Yare ko ay hairakhiye ngakta ngai ko. Yare ko adudi horen reaction center di kadai no hai kanano hai tari chlorophyll A karamba chlorophyll A which is capable of ejecting electron. Electron kapthokpa ngamba jaga do. Reaction start to ngam jaga do ne. Yare ko ana among the accessory pigments kana dum oibe yaiba. Chlorophyll A dum oibe yai tobatabu aduda yawriba chlorophyll A dudi electron kapthokpagi thabakti tawraroi just transferring the energy gi thabak oina tawrani. Yare ko aina ngarang haibada chitharame haina khalamba yai hoji amtang palawrage mayai si amtang ga haibiro reaction center reaction center se accessory pigments na yomjalami se kaigino ngarang haigibani asimadi karigumba chlorophyll A se directly exposed to the sunlight na yaibada photo oxidation toward damage tawi adu prevent tawnabani haina amana ko accessory pigments na yomjalami se kaigino haitardi light of different wavelengths efficiently utilized the way anamne for example sir se chlorophyll A ne lor se yani ko sir se kananom na ingon lupa lising ma pirak trigede shong lodi adai ta tharadi yare ko ana lupa tara pre pushoy mga pre pushoy aina di lor oi di kya su di lo da ana noi na mathang mathang collect halakwan loi khomjal ingon pirakwan you are accessory pigments okay adi 400 nanometer su 500 nanometer su 600 su 550 su loi khomjal na do noi na panel tor chau thoi ingon pisalak to yare ko ai khatang lem tari kya pirak trigede shong lor maroi adumain to na aina harak to se si photosystem chlorophyll A molecule ne muna ta si ko See say light of wavelength kaya no chuksan na ngami se mana 680 nanometer ne man absorbing capacity se yare kwa adubu 500 lakh tar su lokshan gadra di lokshan ni ba kana na lokshan ni kana through de wena accessory pigments ki through de wena lokshan do ne ba sir na hairak tu se khangi kwa ana accessory pigments yorm bai kana ba se light of different wavelengths can be utilized to hanbe awan otherwise as yom the 680 den lokshan ya arwe bro yare kwa okay now see photo system kya ne ba photo system 2 ne on the same thylakoid membrane sida karamba photo systems yoram gane photo system 1 yoram moidra let me draw here the photo system 1 see na photo system 1 gi reaction center chlorophyll a dune lor se see photo system kya ne lor se ba this is photo system 1 yani ko photo system 1 gi reaction center surrounding pigments are called the accessory pigments so yani ko sibo yari bro okay now hozik reaction se kadai they start to get aina talk to shi karamba mechanism na cyclic or non-cyclic in non-cyclic how many photosystems are involved two and we have both the system hozik ready the system anima but a reaction the karamba system dagi hoge have a kanda photosystem 2 dagi howe yare ko ana photosystem 1 and photosystem 2 haina tholi se it is not in order of reaction ate it is in order of discovery ni hanam su even event crt da yare ko reaction di kai de honi ba reaction holagi unko hoze light of wavelength kaya na activate ton sige 680 nanometer la eba this is 680 nanometer wavelength of light through the accessory pigments they strike upon chlorophyll a reaction center sila tare yani ko hoze chlorophyll a molecules na do chlorophyll a molecules absorb light light is energy right energy absorb to ko any atom mana or molecule mana kai absorb to tarde energy absorb to tarde that atom will get excited electron excitation to entam bodo chemistry da khangi ko for example ai noi nungai na ba machin tong ulak ke ko okay mayai da kayaw do atom gi nucleus this is k cell l and m ne ye da kaya lay do electron ani 1 2 3 4 5 6 7 8 1 2 what atom I'm drawing magnesium ne atomic number 12 yare ko sidai sida klm cell ohum sida nucleus lom da changjali gai energy wang dora nem dora nucleus lom da changjali gai k ga m ga kanana energy state wang ge m na lapthui gai wang nucleus lom da changjali gai energy state che ke do ba low energy state ne ba khangro sir na ise adu oi ba taragadi hozik aina hairak to sa hozik k da leiba electron amana energy absorb tore it is gaining energy. Energy wang kale ko there. Only then it can jump to the higher energy state to in chung kani badung kan. Hangro sir say when an electron is absorbing energy, only then it can jump to the higher energy state. Yare ko. Likewise, akhoi ki case sida somda amtang yengak siko ai noina nahan ta uphill downhill me amdo palak trobani. Chlorophyll A is absorbing energy as a result aduna maram oira kanda chlorophyll A molecules the yorm electron ama phakse kapthore eject out jump to higher energy state ta chongi haineba yani ko kanadagi electron kapthore ba adi in other word yam samtakna haragana k cell da leramba electron ama kanagi chlorophyll A ge now jump to higher energy state am da chongkhle phangro k na hairagum se duri atom amgi manung da wen chat khidore ina haining isi chlorophyll A reaction center of the photosystem 2 shina light of 680 nanometer na tarba matamda mangondagi electron pair eject tore bedona ba higher energy state ta paikhle haine rani ko dagi kapthore be electron se it is accepted by kanana okay have matamda thiophytin go ba electron acceptor amak noe ba kanana thiophytin muna tau ko hairiba thiophytin sisu kadai waida dum leiba structure no thylakoid ki membrane da dum leiba structure ngasai sir na both se kai na ge all the reaction happens on the thylakoid membrane only yare ko to batabu thiophytin se kadai da leiba di ormane higher energy state da leiba acceptor ne base khangi ko awang ba state ta leiba hai tare sir do makha dagi si ta na ka khalak ai tare energy drink khara thak ta di ka khalak ya goiba ani ko energy do gain torakanda higher da chongak to ne ba kumta ge adi energy um kedrom tare on pithom tare khambro sir nai se hangi ko low dagi high da chongal ka der energy absorb tor chongal le high dai low dong kumdra ka na energy on release tor kani hangi ko hojik te khoi chongkhat pe wari ra chongthabai wari ra chongkhatna ba gidamakta chlorophyll A is absorbing energy. Only then a pair of electron is ejected out, jump to higher energy state. At the higher energy state, it is accepted by an electron acceptor called as phosphorylated acceptor. Yeah, reverse ibogta. I simplify the explanation ama han tawgi ko. Adong hale thala kot membrane to yam explanation am chong tawsil. See kangi ko. Now, from the phosphorylated sidagine, the electron is then passed down. Electrons passed out thare. Phosphorylated oe, phosphorylated o matungda. From the phosphorylated, electron is passed through another electron carrier called plastoquinone. Kaya plastoquinone the pitare. Kanadagi phosphorylated dagi. Yare ko. Now, from the plastoquinone, the electron again pass to Canada cytochrome B6F complex. Ko electron carrier no pila cytochrome B6F complex. Electron carrier ngakta ko. Sidai kenom hai geba electron transport hai se kemtana te ko. Electron se mathang manaw na electron transport to ge hai tardi kamaina electron transport se to ge hai tardi through the process of oxidation and reduction. Yare ko. Example on pigem ko. Aina nangonda electron am pirega. I'm saying set to be oxidized or reduced. Oxidize electron loss to ine bengonde. You are set to be nang na thangnab du nangon nang electron no pithokreba. You are now again oxidized thangnab do mu. Now, the chlorophyll A, what is happening to the chlorophyll A right now? Oxidation. Phosphorylated is reduced. Phosphorylated is oxidized. Plastoquinone is. So, through the process of oxidation and reduction, electron is passed one after the another. Matang manaw na ina passed out. Yarni ko aduri redox reaction electron transport ki matam da thok toise. Khangi ko matang pithare hojik basic ang pay thoke ko. Okay. Yung ko sir. Kay no piray si electron ay sway ng hatang thamgi ko. Now, cytochrome B6F complex dagi amuk mathangi electron carrier the period. Kanano nga side sir na piba haina hairisa electron. Electron se mathangi electron carrier si maming plastocyanin haina ko. Kay ko ba? Plastocyanin. Kanade thokiba electron nga side travel to risa. Chlorophyll A they kapthokiba electron do aham the phosphorylated away. Then from the phosphorylated it travels passing through different electron carrier which are also located onto the thylakoid membrane. Yarni ko power la em. Yari ko ngasai sida kakhat krizi si low dagi high da chongum ni ko. Awang mo leire ko. Kasi awang mo leire mo de kubhare eba kay thadro ne. Energy is released because this movement is downhill ko. This movement is uphill ko. Yani ko awang mo da chonggalak pa se awang mo dagi hojik se mo kay dawre ine electron se travels down the gradient. Amu kubhare eba. Kay thado mani aduda thadorokpa energy do utilize tawre kanada ADP amaga inorganic phosphate amga chakhai na hore and prepared ATP prepared to one. Congress and I say the energy released during electron transport is used for the preparation of ATP. I should my high here exactly na triko hana basic amtang khangi se sibo yari ba mayam ham gige ngaktang sila matang swaida same thylakoid membrane sida photosystem 1 yaurira photosystem 1 dasu light kya na activate toramni light of 700 nanometer taramoidra ne maduri masu energy absorb toramni na mangondagi electron pair ama eject tore yare ko to higher energy state chonge mangondagi thadorpa electron se sina it is accepted by ferredoxin go electron acceptor am no kanana ferredoxin yari bro sibokta yari ko hoje ferredoxin is set to undergo reduction ferredoxin nagi finally electron se kanada pigige have a kanda electron is finally given to NADP plus the p yani ko electron gain total NADP plus is set to undergo reduction NADP reduction tawgedi kai swap chalamani NADP has to the reduced away hierarchy Kangi bremo yari ko sibokta now loidore munatasi sidai kai kapthokre electron aduri sida electron deficiency am laire ne adui electron deficiency do lady gai dip light phanam tar su makha ta ba electron ka thoro ya roi ba until unless deficiency do kai dodri gai di mention dri gai di sida lairamba electron awat pa do ngasai som dei lakpa electron do na mention gi wan sida electron khambro the electron coming from photosystem 2 se je kana da pigre photosystem 1 da pigre khangi ba hozik photosystem 2 se electron deficient oibra si electron se si reaction center se ꯃꯔꯝꯗꯤ ꯁꯤ ꯀꯥꯞꯊꯣꯛꯈ꯭ꯔꯕꯅꯤꯅ ꯍꯥꯟꯅ ꯃꯈꯥ ꯇꯥꯕ 680 ꯅꯦꯅꯣꯃꯤꯇꯔꯅ ꯐꯅꯌꯥꯝꯅ ꯐꯨ ꯇꯥꯔꯁꯨ ꯏꯂꯦꯛꯇ꯭ꯔꯣꯟ ꯊꯥꯗꯣꯔꯛꯎ ꯌꯥꯔꯣꯏꯕ until and unless ꯑꯗꯨꯒꯤ electron deficiency ꯗꯨꯃ make up ꯇꯧꯗ꯭ꯔꯤꯈꯩꯗꯤ ꯑꯗꯨꯅ photo system 2 ꯁꯤꯒꯤ ꯃꯄꯥꯗꯀꯣ ꯀꯩꯗꯧꯒꯦ ꯍꯥꯏꯕ ꯀꯥꯟꯗ water ꯁꯦ ꯊꯨꯒꯥꯏꯕ water ꯁꯦ ꯀꯩꯗ ꯊꯨꯒꯥꯏꯔꯤꯕ 2 proton ꯌꯥꯔꯦꯀꯣ hydrogen ani ꯌꯥꯎꯅꯦ proton ani दा dissociate तौरे plus oxygen valence तौगे ओम खतनीना आई half के ममांगदा याब्रा proton ani pithoradi kaisu pithomgani positive ngakta thadok yabrane yade electron ni su pithomoidra ade thadok pa electron do sida awat pa electron do da pigre khangro sarnai se khangi ko kai jaga do no water thugai se sida water thugai base water dissociate ton base we call it photolysis hane ko kaidoge maram di sigi degradation of the water se it is induced by light in this tube sida light na tabra adai kai kapthoki adi sida kai watle adu wat pe main na bani water kai rise si light na taramdraba sidum electron yaw leiroidra dai tari water do thugai gidra dai tari water kai rise light na induced over reaction ne se ana photolysis of water haine ko yara bro adudi sidai su khange akhoi oxygen se kana dai thadro poirame water dai thadro poirame and the job of water is to keep proton pina bane yarni ko reducing agent when act tobani haina nan hai give do sigi reaction se thoke ai tardi we require an element that is manganese kai dakar hoiba manganese dakar hoib yare ko ai sina loiba natri ko sidi ai basic amtang hana chawap amtang khangan gen takpane amta tabiro loidore adi water dakar oi se kai gi oile me electron pe adi if we keep supplying water water will keep donating electron si electron se piri gai si cycle se dum tawle ni as long as this flow is there what will keep on producing ATP will keep on producing and NADPH2 will keep on producing and also oxygen will be continuously released oxygen adum thado le ni haidone yare ko photosystem 2 dagi chatkhiba electron si photosystem 2 da mo halakra amo halaktre so this is called non-cyclic photophosphorylation haina ko yare ko khangdaba yaram gini ngakta ngai ko ai NCERT duga mana no explain troki sida ai khar sarna noi gyan taba line ba na kitang twist story yare ko aduri somdam tem ko reaction kai thewe photosystem 2 dagi electron thole ko higher energy state ta chongkhaleira kana no e adai de electron dom kumthara ba ai ko ise downhill photosystem kya da tare adai mu kakhala ba the movement of electron follows jet-like structure hence it is called jet scheme haina ko yani ko electron chatpagi pathway se jet man breng shape aduna si kai ko ba jet scheme haina su khangne ba yani ko karamba mechanism se non cyclic photophosphorylation yarage cyclic tumtanga sir comparison tauge aduga si sai hoji details se lakhini ko sine yagiroi so what are produced during non cyclic photophosphorylation do we have ATP do we have NADPH2 and do we have oxygen? Yes, so the products of non-cyclic photophosphorylation are ATP, NADPH2, and oxygen. Yani for the production of oxygen, we need photolysis of, for photolysis of water, we need an element called as manganese. Darkar oi, habitumaga su khangjan Yarage. Okay, next. Somdemo. Handak kisina kay muktak to ba? Cyclic photophosphorylation. Kabak niko. Alo ngale ay details ta poroge. Handak kisina cyclic photophosphorylation ne. Ngasai da sarga wari sanaba kanda cyclic photophosphorylation di photosystem kaya khaktamak involved tawi haige. Photosystem amakhak involved tawi haige ko. Karamba photosystem? Photosystem 1. Ay tari sir sila tharage. Photosystem gi mayay da kay leramgani chlorophyll A karamba photosystem ki reaction center no photosystem 1 akoi bada su kai leramgani accessory pigments leramgani so let's say these are the accessory pigments ne lorsa yare ko now hozik light kya na activate to dono so light of 700 nanometer comes and activate chlorophyll A okay adoi tari chlorophyll A de kai kapthorani chlorophyll A dagi a pair of electron is released and jump to higher energy state laeba ko ngasai photosystem 2 de yana na kapthokiba electron do kanan okay. So please remember, in non-cyclic photophosphorylation, the first electron acceptor is pheophytin. Whereas in cyclic photophosphorylation, the ion and the electron acceptor se kanano is yet to be discovered. Kangdri. Aduna, I will simply put it primary electron acceptor. Main acceptor amana oe katang harameba. Yareko, primary electron acceptor amna accept tawre. Adi primary electron acceptor is now set to undergo reduction. Now, mangondagine, how is the electrons at? The electron is now coming down the hill. Kumda gaure, awang bade ani mada kumda gaure. Like M the K rom da mulak ba hore hani. Yore ko electron hariba electron se mathang kana phawra lake da sidana nahan bada kana phawge da ferry doxin na hana lay sidana. Yare ko ferridoxin gi mathang dagi cytochrome lay cytochrome katang harage B6 ka haparoige yam important nate cytochrome dagi kanamu phawre kananu ngasai de phawre sibo electron through the process of oxidation and reduction, right? Sidagi kanada plastocyanin dong lae hang yai ko ngai ko soire I shall start with it should be plastocyanin kainom chenari ba malida plastocyanin sinakai peridoxin kainom chena ba male service like tadi direct yaudam yeah look here I sem dro king aiko asi yori ba components na te ba darkar se a mechanism duneva kaino move to electron is moving uphill or downhill electron si makha da kumdara di kai thadongani adugi energy ro shenaraga ATP amaga inorganic phosphate amga chakhai ne hore aduga kai sungeba ATP sungeba hoji da electron what libra electron what are the makha ta electron thadoro ya roine adugi awat pa electron do asai chat kiba same electron no mahal mention giban aniko so the electron moves in cycle as long as this cycle is going on it will keep producing ATP dum sungle ni ana masamak gi kapthokiba electron sa same sentence da mo halai haidana sina cyclic photophosphorylation haina ko why called photophosphorylation because both involving preparation of ATP phosphorylation adi ATP sungbase anima ATP loi sungeba amana cyclic manner da sungba amana non cyclic manner da sungba khatane yare ko kai khane here how many photosystem involved here only what is produced only ATP is produced. Whereas in non-cyclic, ATP, NADPH2, and oxygen. Huma putwe. Yare ko. Hauzik sir noi basic sima chorapang khangam ni na ko. Now let's go deeper. Kare lun chak chalase. Yare ko. Yango sida mayam. What am I drawing here? Chloroplast. What is this individual? I thylakoid ama lokthoroge ko. I'm going to take one thylakoid out and I leave a thylakoid to. Sir, swai de lake. So thylakoid amase. Let us say this is one thylakoid. Nahan suye kram niko. Thylakoid si membrane kaya na surround tawi. Membrane amakhatani ko. But why am I drawing two? Because is one is representing lipid nebacity. Adi lipid ki layer ani suya hai neba. Yare ko. The inner cavity space is called lumen. Very good. Yare ge. Yare ko. Sida lipid nila ito som the lipid bilayer lipid, som the lipid smile ito. Yare ko. Si se hauzik sir zoom out to game ko. Khar chong ni se asumai na di aro. Ay achauba thela ko ram yela ge som the tabiro. Just a second. Okay. Let's say this is thela koi. Kitang uniform o hen ge ko. Swai shile na PN mani thoi na. Okay, si thala ko ni haina lower sip. Yare ko. Adoy tari, what is this? This is lumen. Yare ko. Som sina, auren, som sina inner lipid bilayer layer duni, som sina outer ki lipid layer duni. Yare ko. Okay, hafsal loige. Hairiba thala ko ki membrane se lipid bilayer se. Sidai kana embed torlay dona. Photosystem sing electron carrier sing se embed torlay dona. ꯑꯗꯨꯗ photo systemꯁꯦ ꯑꯗꯣꯝ ꯊꯪꯕꯗ embed ꯇꯧꯔꯒ ꯂꯩꯒꯦ ꯍꯥꯏꯕ ꯃꯇꯝꯗ photosystems are found embedded onto the inner lipid layer. ꯃꯅꯨꯡ ꯊꯪꯕ lipidꯀꯤ layer ꯔꯣꯝꯗ ꯂꯩꯌꯦ. ꯀꯗꯣꯝꯗ ꯂꯩꯌꯦꯕ ꯃꯌꯥꯝ? Okay, ꯁꯤꯁꯦ ꯑꯩ ꯃꯆꯨ ꯑꯉꯥꯟꯕ ꯑꯃꯅ ꯏꯍꯧꯒꯦꯀꯣ. ꯑꯩ ꯍꯣꯔꯦꯟ ꯅꯨꯡꯉꯥꯏꯅꯕ. ꯃꯅꯨꯡꯂꯣꯝꯒꯤ lipid layerꯗ ꯑꯣꯏꯅ ꯂꯩ ꯍꯥꯏꯕꯁꯤꯃ ꯀꯥꯎꯕꯤꯔꯅꯨ. ꯁꯔꯅ ꯁꯤꯗ ꯌꯤꯁꯦ ꯀꯩꯅꯦꯕ? I'm drawing photosystemꯅꯦ. Photosystem 2. Okay? ꯑꯗꯨꯗꯤ photosystem 2 ꯑꯣꯏꯇꯥꯔꯗꯤ ꯃꯌꯥꯏꯗ ꯀꯩ ꯌꯥꯎꯔꯝꯒꯅꯤ? ꯃꯌꯥꯏꯗ reaction center chlorophyll. So this is chlorophyll A. ꯀꯅꯥꯒꯤ chlorophyll A? Photosystem 2 ꯒꯤ chlorophyll A ꯅꯦ. ꯌꯥꯔꯅꯤꯀꯣ? ꯀꯥꯏꯋꯥꯏꯗꯅꯣ ꯌꯤꯁꯦ? ꯊꯥꯏꯂꯥꯀꯣꯏꯗꯀꯤ membrane. membrane ꯒꯤ lipid layer ꯗꯨꯗ. ꯃꯅꯨꯡ ꯊꯪꯕ lipid ꯗ. ꯌꯥꯔꯦꯀꯣ? Similarly, ꯀꯩꯁꯨ ꯌꯥꯎꯔꯝꯒꯅꯤ? Photosystem kyasu yaramgani? Photosystem 1 su yaramgani. Let's say this is photosystem 1. Photosystem 1 adi si pigments ama khaktang ni natra. Natti group of pigments neba. Mayayda kai leibo ano? Mayayda chlorophyll A reaction center leibo ane. See chlorophyll A dune laurasi. This is chlorophyll A reaction center, but for photosystem 1. This is photosystem 2. Yare ko? Howzik light kya na activate tawduna photo fare no yengba nungaina ba this is I'm going to put this is photosystem 2 and this is photosystem 1 adusu thousand birage sir yare ko see light kya na activate reaction should start from which photosystem 2 dagi light kya tara ani sida now light of wavelength 680 nanometer strikes upon reaction center chlorophyll A. As a result, chlorophyll A molecule they kai kapthorani. Chlorophyll A molecule, because of the excitation, now ejects a pair of electron. Electron pair ama kapthore. Khangi brahayo? Khangi ko? Masida thadori be electron se kanano oktoino. It should be accepted by pheophytin. Chumiko mayam, hairi ba pheophytin do. Ma di kadom da lege hamatamda thylakoid membrane ki outer lipid layer om da layer. Yani ko? So I'm going to put here, what is this? What is this? This is pheophytin. Pheophytin. Yarabro, sarna taisi yaribro. Yariko, hozik pheophytin na accept taurani kanase that electron do. Okay. Yariko. Now, from the pheophytin the electron is then passed through another electron carrier. Amakakta passed away. Kana oigi ngasai? Kana oigi? Plus 2 quinone. Let's say this is plus 2 quinone, sinelorsin. Munatasika. Kanada pido revozik. Electron se, electron is given to plus 2 quinone. PQ stands for plus 2 quinone. Plus 2 quinone se, electron lower matamda, ma kari su lower ngamge am talako. While taking electron from pheophytin, it also take a pair of proton juram solchaleva kadom dagi. What is this side? This reason called stroma. Very good. Thela ko electari mai mapandri kay koromani. Stroma koromoidra. Let me put this side is stroma. Yare ko? Plus 2 quinone di electron puwa khakta natana kay sulawang ngame haina. Proton sulawang ngame. While taking electron from pheophytin, it also take proton from stroma. Stroma de laure ko? Laura ba matungda kay ole it get changed into kay da change together plus 2 quinone hydrogen si ole. Yare ko? PQH2 high structure se kari yauraba structure na it has got 2 electron and it has got 2 proton. Proton ani yauraba electron ani yauraba structure ne batudi. Hangabo. Now from this PQH2 se sidagi electron is again given to kanada pireva to cytochrome B6 F complex the pirev. Can I put here cytochrome B6 F complex the period. Khangi hero? Yare ko? Electron peak raba? Ngasai electron lo wada kai shulawi? Proton shulawi? Hoje electron pithodi kai shu pithokane? Kai? Proton su pibam thoktra while giving the electron out, it also give kai proton kadai by the pishalameva into the lumen. Lumen si the proton su pishalameva. Hangro sir na say yariko sibogda. Okay. Adoi tardi ay marak amtang thalaw gige sige movement se sum lady gai di kai sum pushalakane. Proton se lumen da sum pushalakone horin chemiosmotic hypothesis su yansel lorge to ne sir swaida. Yariko. Now, hozik kanada lady electron cytochrome from the cytochrome electron is then given to plastocyanin. No period. Kanada pireba plastocyanin. I'm going to put PC here. Yanika hozi focus test da plastocyanin se kai thui reduction thui ba ai sibok tang hak tang post ta thamgi yaraba somdai lakpai lambi se yareka matang swai shida photosystem 1 chlorophyll a se ju kanana activate swai torongane light of wavelength Haibiro light of wavelength 700 nanometer na masu ngasai ni strike tor thama maroidra ne adi mangonde kai kaptho mani sidai shu electron pair ama kapthoeba yare ko hidai kapthori be electron se kanana okhi ningsangi bra accepted by ferridoxin oeba yare ko kanana neba ferridoxin FD stands for ferridoxin which is located towards the stromal site stromarum the layer hangi yare ko now loidore mati ang katang ayre ko ay sidam tang hapsal hugi gyong okay here machu se mana da bam hap ke ko kitang mun ta su ko sima NEET ta hangre hoje ye leisi aina sida pink machu na yexali se ay hana level toge then I will do the explanation this is called NADP reductase enzyme hene ko kai ko ba si enzyme si maming NADP reductase hene ko NADP kai reductase now hoje hoje electron kanada yauri electron kanada yauri ferridoxin da yauri ba electron se it is finally given to kanada pireva NADP plus the pireva nicotinamide adenine dinucleotide phosphate plus adi kai wat hai na electron awat pa electron do pibireva so that it will get reduced. But for this reduction to happen, we require an enzyme called NADP reductase, which is always present on which side? Lumen side or stromal side? So, stromal side yawe, kadom side yawge hangakrib. Yareko, si kai form tawni duri, what is happening here in this reaction? Reduction. For that reduction, we do require this enzyme called NADP reductase, so that it will get reduced into NADP kai raise to the reduced tore na haidone. Khangi ba? Khangi bro? Loidri ko matat yam tatli matat me amsam ta samjan gi ge kanadi kapthorak pa electrono. Photosystem? Adi photosystem wan se electron wat ine. Mangon dagi wat pa electron do ngasai shidagi lakiwe electron do na mention gre haina hairak to ne. Kanadi lakiwe electron photosystem 2 dai chatkiwe electron do na. Asi problem laitre hoje. Yari bro? Yari ko? Photosystem 2 gi oina di still kai wat le. Electron deficient oi maram di mong de electron ani mo already thado lai khram ni na adi anyhow we have to manage the deficiency do men hana mention da di aroi ba adu adugi deficiency do mention na bagi damakta photosystem 2 mapa sida kai thugai bo ano water say photolyze I'm going to write H2O is degraded into two proton and half oxygen sing plus 2 kai plus 2 electron amne ba electron do shidong ulage sila kai pi khani electron do som da pi gre haine ba khangra sar nai se khangi ko adoi tari water hai basi lai ri kai di water na dong kai pi ne electron ani ni photosystem kya da as long as water is providing electron to the photosystem 2 will keep supplying the electron to photosystem 1 and the reaction will go on adom jatha le ni yani ko adoi tari kai dum sung lai ne ATP su sung do ne ba ATP se sar na nga chin haraga swine flu so we ka do mai ya de bai huji mechanism hairage sido ushan ge roige heba yari bro yari hayo what is this reaction called photolysis of water for this reaction to happen, we require manganese. Kay darkar oiba? Manganese darkar oy simtanga kaubirano. Sibo yariko? No. Adoi tardi sumaina sum move tawrigaidi kay sum changakani lumenta. Ngasai plus 2 quinone sum pusilapado. After a while, accumulate sum tawrani kay accumulate tawrani hydrogen ice plus ice plus ice plus ice plus sum accumulate tawre. Yareko? Now, question hangye. Lumenta ice plus angle is kay nanon. Asai PQ na pusilapama? Was ngahalay tan water splitting sum tawlay tari sidasu kay tadoke. Two ice plus every time when water get dissociate, it always release two proton. Chumbra, and there are two factors that are responsible for increasing proton concentration in the lumen. One is PQ na pusilapama, amana water splitting tawama. Yareko? Somday kay handarakani. Proton handarakay maram ani hero. Amana, ngasay PQ duna proton lagthokpama, somda pump tawbagi, amana proton se kay the use tawthuye. Proton is used in reduction of NADP plus into NADP haste to the reduced over the use of 12. Aduna stroma romdana kay shumandare. Proton lumen lomdana proton sum hengalev. And after a while there develops a proton gradient across the thylakoid membrane. Thylakoid membrane yamna khetna proton ki gradient nama develop tore. This side high or low, high proton concentration, and this side low proton concentration ama generate tore haraktone. Aduri yam khenarak tardi somti wange. Pitrivida leiba posak khudingma awang bade anemada loi cheningi. Tra adu hoji proton se kadom yam thomninga mane. The proton has got a strong tendency to move out of the lumen into the but why? Because thylakoid membrane is impermeable to proton. Thylakoid ki membrane se chatpayadeva kanada. Thylakoid membrane se proton phawayadeva. Adi thoki thokninga bane bayamna. Yarani ko. Adi sumaina kamaina thokani haina lairing aisada a hero comes into play. And that is none other than ATP synthase. Thylakoid ki membrane da sukai na peba ATP synthase na peba. What is this? I'm going to write this is ATP synthase. Yari bro mayam. Yari ko ATP synthase na haiduneba. Nang yam thutni do. ATP synthase is something like get. Yani ko ay power thokso. Toto you have to do something for me. Ay kainom di tom da yaro isine. Yani ko horen ma power thok matam da ATP mam sungbiram do. Kamay sungi hierarchy ngai ko. Yari ba sibok kay never say no. Hydrogen when urakano ba say. Kay na wangi say hydrogen. Ising when uwa. Awang bising somsina anemba. Hoje awang baday anemba de sing selan seva. Marak today turbine am tamban. That turbine is none other than ATP synthase. So when water pass down, the turbine will get rotated during the mechanical motion ATP sumo to neba horen. Kang bro. Sida kay mayam naire. Proton. Now the protons start moving through kana formula eba. ATP synthase formula. When it moves through the ATP synthase keep rotated. Adugi mechanical motion duda ADP amaga inorganic phosphate amaga chakhainaraga ATP sungi haidone. Yarabo, what am I showing here? Non-cyclic photophosphorylation, and also I'm showing how ATP is prepared during non-cyclic photophosphorylation. This mechanism is called chemiosmotic hypothesis. Yandara dro. Kara lubro. Ude tilai. Yareko. Sir, amka hierarchy munato. Amka hierarchy gave. So I the kitangka luba hierarchy. Luba dinate ko. Simba pudweni. Like NADP reductase kadom de yawi. Ko. Like pheophytin kadom de yawi. Water splitting kai jaga da tawi. 1 da tawra 2 da tawra. Water splitting da kai dakar oi. Yani ko non-cyclic ki production kai no. Cyclic ki na kai no. Kai na to swai sikatane ako na fathers. Yareko. So whether it is cyclic or non-cyclic, what are responsible for preparing energy, right? In the form of ATP ne. Yareko. So what the mechanism se kitang khena bane. Let's recall the non-cyclic photophosphorylation to how many photosystems are involved. We require two pigment system photosystem 1 as well as photosystem 2. But reaction start from. So when that photosystem 2 is struck by 680 nanometer, the reaction center chlorophyll A molecule ejects a pair of electron, which is accepted by, which is at higher or lower energy state. Pheophytin from the pheophytin, electron is passed to plus 2 quinone. But there is one problem. For plastoquinone to accept an electron pair, it also has to accept a proton. I'm slow bomb change. Kadom site. The plastoquinone stroma rom de proton loctave, thereby reducing the concentration of proton in the stroma. Loctave ko, yaraba, the plastoquinone undergo reduction. Reduction right into plus PQH2 dole. Plastoquinone de mukanada pire electron do cytochrome do pire. Yani ko electron do piba matamda mana nga sai pusalakiba proton do kadai da langsalameva lumen ta langsalame. Yaradra. Now from the cytochrome electron is again passed to plastocyanin. Swibok tang thamgite yaro. Marak horin kon samdoine. Ere nga sari lakpe lambi mapung phadri plastocyanin de lephali ko. Matang swaida same thylakoid ki membrane du da leiba photosystem 1 du se activate toro moidrane. Light wavelength 700 nanometer na mangonde kaishu kapthongani a pair of electron accepted by ferridoxin. Yani ko ferridoxin then pass the electron to NADP plus the period. Adom diawe. Stroma. Yani ko NADP plus now get reduced to NADPH2, but for this reaction to happen, we require an enzyme called NADP reductase, which are found attached on the thylakoid membrane, but towards the stromal side. Thylakoid ki membrane dadi dum yaujeba, but stroma ki side lom doino onjhala lei wanida. Yare ko kapthokre photosystem 1 dage electron. The photosystem 1 is now having an electron deficiency. That deficiency is now kanana mentioning greva. Photosystem 2 de lake plastocyanin de pira mention grave. Yare ko songi prom kyam leitre songi. But photosystem 2 is still electron deficiency lady. Yani ko adi photosystem 2 the lady electron hole duma mention gaya tadi here we need photolysis of water. Water photolysis the car wave. Yare ko lysis side water glycolysis tarabra kai thugai bena glucose thugai be lysis adi breakdown heba. Yare ko adaina ayek to photolysis sai sida amu lana chatkini ko water light na thugai ra si water se natteba light na induced over reaction haitare kai do no si thugai ri se electron p na ba kanada PS1 do PS2 do kai electron deficient oi be kai gi deficient oi ba light na taru bai light taram dadi deficient oi roi ne adi si kai roi haine ba sir na hai se light induced degradation of the water ne ba hence it is called photolysis and we require manganese for this reaction to occur yare ko electron p graba the reaction continues as long as aduri si gi reaction se dum thoi gai di proton se dum pushalok tori hai ba kai de stroma rom dagi lumen lomda lumen ta su kai dum thugai dore water every time water thuai mateng thugai bu kuting kai dum puthui si da so adi proton sum accumulate tore lumen manung sida as a result after a while the concentration of the proton ti wang khlani kai manung da asom na sum handarak to stroma da handarak pai maram ani pi ba direct ta pq na lok thokhi be ma pam to khi bai yare ko amana reduction the use to thokhi ba NADP do reduce to wala yare ko asom na handare somna wangale aduna maram orakanda proton gi gradient ama develop tore across the thylakoid membrane da yani ko gradient in the sense that concentration canare high concentration in the lumen low concentration in the stroma yani ko concentration kenarak tardi awang borom de ane madong chatninge kadom da lumen tagi stroma da chatke to bakan se chatpa ya de proton je maram di thylakoid ki membrane se proton da impermeable oye aduri kamaina chatpa ya ge haina thirakpa kansa thongam palem done ATP synthesis ko thongama ATP synthesis na power chatlan ban yani ko power da mana x amam la ho ATP amam yare ko ana proton haina khanganu water na move taraini here ATP synthesis takes the role of turbine urungani adugi mechanical motion dura kana sakhaina hoeba ATP amaga inorganic phosphate amaga aduga ATP sungi na haido sigi mechanism se kai koeba chemiosmotic hypothesis of Peter Mitchell koeba yare ko aduri ai ngasi noida hangdoisa photosynthesis da chloroplast nung the energy sungbra kari chloroplast energy sungbro sungi adoi tari energy sungbo organelle ani yawe cell gi manungda nahangai ama khakta na ti not only mitochondria chloroplast is also involved in preparation of energy not just food khakta na ti do we have ATP now in the second step we are going to use this ATP and prepare glucose that very step is called as aduna dark reaction da talak to hangadro sibokda yare ko topic kan taisa topic humni ko ai khara samnaba haina I'm talking about non-cyclic photophosphorylation cyclic photophosphorylation and I'm Also explaining the mechanism how ATP is prepared during non-cyclic. We call it as chemiosmotic hypothesis. Ahumneba samdatlise. Yarage, aina khanbada sir loima pallamneba. Ko ai sir gamtang pass yung hojik. Ado gyan darni. Ai cyclic to the yaradro. Cyclic city am lai. Why is it called cyclic? Because the electron released from PS1 returns to the same PS1 again. Amo halakpay neba. Yani ko, kay kagtang di shungeba sidana. Sidana ATP kagtang shungdoba. Adubo ay kwena taiba chemiosmosis sidik kay dadi best or tayo. Non-cyclic tani ko. Cyclic su thokti thueba. Ado like tayo shiri non-cyclic to kayo. Yarage? Slide change okay? Okay, look here. Si pabiyo ko. Wari su nungai ni pabiyo. Ay gadi paarwe. Nungai gani Kelvin gi wari ni. Melvin Kelvin. Mazidi yam thi thagat ningai ni ko. Yam thi luba reaction hai mana explain to misi Kelvin cycles. Yare ko. Ay hayeng thi nungai ni hayeng wari to roke mana kamai na tau gi gel. Yam kuin jang gi ma. Hayeng tau se si wari sina. Hoje ay koi ko. We discuss about the absorption spectrum, the action spectrum. We discuss about chloroplast. We discuss about various pigments. Mayama takrabani. Now I will start from here. Swaide ham samjalema. Paring biro ko. Ay dis de horke. Chemiosmosis de. Yaradi wari du ngaktang sabiraktragadi recording so tauri. Ngasi lakta ko amusong hangaroidam. Ay thagal oiba video. Yan ko. Chemiosmotic. What is chemiosmotic hypothesis? It is hypothesis that explain how ATP is prepared during light reaction. Light reaction. Especially NCRT dana karamba light reaction the best or explain to wa. Non-cyclic explain to jabaneva. Light reaction gi matamda ATP se actually kamai prepare to wi haina tapa mechanism do hypothesis to. Adu neva chemiosmotic hypothesis to. Now let's see. Let us now try and understand how actually ATP is synthesized in chloroplast. Yani ko. So from here, we came to know that it is not only mitochondria which is capable of preparing ATP, chloroplast is also capable of preparing ATP. The chemiosmotic hypothesis has been put forward to explain the mechanism of ATP synthesis. Like in respiration is the way we go. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of proton gradient. ATP sungeti proton ki gradient ama hana generate to handa diyade. Yani ko proton ki concentration khenaba ama hana semgatra diyade haini. Yani ko across the membrane, akoi ki case da kai membrane. Thylakoid membrane. Muna tau ko hayeng noi di medical thadoni na entire syllabus to parak na. Ay silorga respiration lerem do male. Ko respiration paba kanda comparison to wada horen mitochondria across the kai membrane no. Hojik te itakiro. Horen mitochondria across the inner mitochondrial membrane when hierarchy. Eriko kona kanasi. This time these are the membrane of kai ki membrane neba. Akoi disida dia. Thylakoid ki membrane. Mitochondria dana inner membrane or to. Eriko. There is one difference though. Kena bam dilay. Here the proton accumulation. Proton se katolom de accumulate ore. The proton accumulation is towards the inside of the membrane. That is in kaida lumen. Lumen lomdo in a proton accumulate ore. Horen mitochondria dana ko outer and inner membrane do intermembrane space to adudo in accumulate oni. Yare ko mitochondria comparison to gumse sumayam tabiro. Yaro idori shide yawame. In respiration, proton accumulates in the intermembrane space. So the comparison am tang power si hojiki wene diko. Hojiki wani hapta amatung da yam thi hairani base. Yare ko hojiki wena kai wai da proton accumulate tawreba chloroplasta. Lumenta horen mitochondria dana outer membrane ga inner membrane ga marak to kai koya. Intermembrane space to accumulate tawi duma khangtare. Yare ko ai soidana comparison torokige respiration tapagansi. Of the mitochondria when electron moves through the electron transport system. Yare ko param kanduna proton jisum pusalak to shiwa. electron duna electron carrier mayam do power ga chatpa matam da adugi electron power chatli by system the electron transport system ko khangage yare ko let us understand what causes the proton gradient. By the way, proton gradient developed to across the membrane. We need to consider again the processes that takes place during the activation of electrons and their transport to determine the step that cause a proton gradient. Yani ko, proton gradient se kamay na develop to hay base. Hangge hay tardi electron gi movement. Yani ko, process kamay na start torpi activate torpi hay na chingba chingde khangbam changi sini. Sidai hausi number A. Kayno makhagi points ing talk to say causes for uh, how proton accumulates inside the lumen. Maram singdo. Kan the kay splitting of water kai waida takes place on the inner side of the membrane, that is lumen. The proton or hydrogen ions that are produced by splitting of water accumulates within the lumen of the thylakoid. Ahando maramam laire. Ereko. Aniswa maram mamu yngsiko. The second reason. Look here. Aniswa. As electron pass through the photosystem, the protons are transported across the membrane. Electron na photosystem the lakwa matamda kay electron ko la isa kay lausalawewa protons are being pumped from the stroma into the lumen with the help of plastoquinone or this happened because the primary acceptor of electron what is the primary electron acceptor for non-cyclic Pheophytin is located towards the outer side of the membrane. Side to outer side of the lake do. Yani ko transfer its electron not to an electron carrier but to a hydrogen carrier. I think aside plastoquinone do. Ma the electron except on ni ma carry on it queen at our living am there. Adi ma the kai carrier ne main na hydrogen carrier ne kadai ta na pudona. Stroma dagi lumen ta na pubangamba. Yare ko hence, this molecule removes a proton. Kanano, this molecule, what is this molecule? Plastoquinone removes a proton from the stroma while transporting an electron. Electron duma so transport to viramba matamda kay suloksaleva proton. When this molecule pass on its electron to the electron carrier, what electron carrier? Kanada no pass to dushe cytochrome da. Yani ko on the inner side of the membrane, kanaso lang sa lameva the proton is released into the inner side of the lumen. Lumen to rom da ang proton do lang sila mer. Ay amtang swai kitang simplify taro ke ko. Pheophytin na oyena. Pheophytin de electron se kanada pire. Plastoquinone do oyeba. Yare ko. Plastoquinone de electron se opo matam da plastoquinone stroma de kayam lawe haino. Kay proton kya. Ani a pair of electron lawe. Yare ko. Tulor matung da PQ ay stu do na electron do kanada transport tor me haino. Cytochrome da sorry yes cytochrome da adu to matamda hydrogen na kai lang salami haino adumaina hydral ore yam kem lude yare ko ahum suba maram na kai NADP reductase enzyme sine to hangri se ko hangram ni ko kadom yawe located on ngai kus kai da ba trauma hectare zero ni ko kai yawe site hare kino kai da yawe thylakoid ki membrane da kadom thangba located on the stroma site of the thylakoid membrane yare ko along with electrons that come from the acceptor of the electron of PS1, protons are necessary for reduction of NADP+. NADP+ na kay oye. Proton nga ko. Electron, gain of electron is ko reduction. Ado reduction tawag di electron kaktang natiwa da car say proton su da car oye ba. Yani ko to along with electron that comes from PS1, ay su da car ba. Protons are also necessary for reduction of NADP plus 2 and ADP H2 doon, ma'am, the car wave. Ano ko? This proton, so kaya nilang tuwin ng proton do. This stromata kay sumandarani. So these are the three reasons for causing proton gradient across the thylakoid membrane. Yaraba. Hence, within the chloroplast, proton in the stroma decreases, handare. Yarniko, while in the lumen increases or accumulate. This creates a large proton gradient across the thylakoid membrane as well as measurable decrease in pH in the lumen. Kaya mahingali Okay, kay yam hengale lumen manungda proton. Proton yam hengala pari yam acidic oire haine. Chumbra yam acidic oire padi pH yam low oithre haine. Stroma dana pH hengala mani. Natragana electrochemical gradient ama develop to hai shwe ko. Manung sham positive oire hai shwe abra. Mapana positive 100 hai shwe. Charge ki difference am lai higher so kem kai deba ko. Why are we so interested in the proton gradient? Para kasi yung sila, aside tayo isip, ang wanda mock talk pamandro si line set. This gradient is important because it is the breakdown of this gradient. Gradient se kaya matam na neba kay sumog do siya. Yani ko, singa wang mama animam layeba. Sum cendhara di level ma naraktora. Adu kaya matam kayan gaya iter cendhara ko da mayay de kay hapoba turbine. Adu electricity yung sumog wa neba. Yani ko, because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of the proton across the membrane towards kadom namo halagtono stroma through a transmembrane protein. You remember transmembrane protein? Okay. Sir, na fluid mosaic model. Plasma membrane ki model kappa matamda bilayer is there. Within the bilayer, many proteins are embedded. We call it intrinsic protein. Some are loosely arranged called extrinsic protein. Some intrinsic protein are too large that they traverse both the layer. We call it transmembrane protein or tunnel protein. Adi ATP synthesis masamok se protein neva. Yani ko, kay protein transmembrane protein neva. Transmembrane protein. ATP synthesis kay neva. Masu protein neva dum. Yani ko, channel of the CF0 of the ATP synthesis. Ay kay haino hierarchy sitago. Eh, sorry. Loidore, loidore. Something yung ka. Masay sir, ATP synthesis ki masak to sumay yekidra. Yeki ka. Mangwanda, sina makongi saruk neo. Makongi saruk, so we call it F0 kawi. Sina head part haye, sina F1 ko. Adura ako'y na kanari ba ATP synthesis chloroplast kini na. Ano kaya mahapa? Siya mahapa katane. ATP synthesis is having how many parts? The base and the head. The base is called F0, the head is called F1 kawel. Yare ko. Kadom lai no di tholok to siya. Si base lom ni power tholok to siya na tra. The ATP synthesis enzyme consists of two parts. One called the CF0 is embedded in the thylakoid. As in the thylakoid, the pumale the saruk to. Apalam the lithori saruk to na and forms a transmembrane channel that carries out facilitated diffusion. Ma power cell to diffusion the awang the name the cell hendo. Yani ko of proton across the membrane. The other portion is called F1 and protrudes onto the outer surface of the thylakoid membrane on the side that faces the stroma. As in the stroma rom the littoral makoki saruk to na. Ito kinga side. The breakdown of the gradient provides enough energy, ado thugay matamda energy am thadore, which is used for kay tawa hayo conformational chains. Karamba part. Shape 1 is ang kambi ko. Conformational chains ay aina tapode ungi haydo. Adi chains so ini. Yani ko adi matamda kay sumugdo ba synthesis several molecules of energy pack ATP sumugdo ne. Yani ko this is called chemiosmotic hypothesis ko. Hayrani da. Yara bro siba wada. Topic kay loire harmo. Kay cyclic, non-cyclic, and chemiosmotic hypothesis.